We're here winding up the CROI interviews. We're here with Romas Geleziunas, who is Director of Clinical Virology at Gilead Sciences. And we really are appreciative of you being here because, as we discussed, there is a sense of getting the right message out, getting the information out that's helpful to get people to understand some of the aspects to what we call the cure and what may be the cure, which we're kind of going down a very complicated path. So before we get started, why don't you kind of give a background on the work that you've been doing and what's gotten you to the point of some hope?
Yeah, so at Gilead Sciences, we established an HIV cure eradication program about 4 years ago now, and we were one of the first companies into this space, and it's been a very interesting journey. This is early drug discovery efforts, so a lot of trial and error, a lot of things that don't work, but we have focused a lot of our attention on trying to identify small molecules that activate the latent reservoir, and this is believed by most of the field to be one of the principal things that we need to accomplish, that is to reawaken that latent virus in hopes that these cells become exposed to the immune system and either with the help of the immune system or other modalities hopefully eliminate or kill those reservoir cells and then determine whether this will ultimately achieve a functional cure. So a lot of our effort was spent on trying to identify such molecules. We've devised and put a lot of effort into this, devising assays, screening, doing high-throughput screening to identify small molecules that activate latent HIV. And we've discovered a few interesting molecules. And one that has emerged from this screen is a drug called romidepsin. The brand name is Istodax. And it proved to be a very robust and a very potent inducer of latent HIV. And we've done a lot of work to confirm those data. And we then decided to collaborate with the ACTG, the AIDS Clinical Trials Group. We reviewed the data together, and they were interested in these findings as well. And as a result, they decided to initiate a clinical study to test this drug romidepsin in HIV-infected subjects on suppressive antiviral therapy. And so the clinical protocol is under development, and if everything goes well, perhaps later in the year, we will initiate this study. And this is going to be very important to us because we really don't know very much about this area, but it's important to establish a clinical test of concept as to whether these drugs that we're discovering or compounds that we're discovering in vitro in our laboratories, they actually translate to activation of HIV in individuals, and they have to do this safely, of course. So that's kind of the first order of business, whether a potent compound discovered in the laboratories through high-throughput screening that activates latent HIV from the cells of patients, would that now translate to in vivo or activation in patients. So a very important milestone that we need to achieve. If that happens, then that tells us that our assays, our tools in the laboratory are good, that they have the ability to identify agents of interest. So it's very important that we achieve this first milestone. As you probably know, Fred, there's been a lot done on SAHA, a drug of the same class as romidepsin. These are HDAC inhibitors. And David Margolis from the UNC has done a very nice study, and he has shown in a group of 8 patients that SAHA does activate transcription of HIV in resting T cells. So romidepsin is a drug in the same class, and so it'd be important to verify whether a drug of the same class does the same thing, or if not, then we would pursue perhaps different avenues. But it's important to verify his findings with a different class of molecule, or a different molecule from the same class, I should say. They're structurally very different. This is why I refer to them as different classes. So this is a very important objective. I know that Sharon Lewin just presented also some very nice data here at CROI, and again showing that SAHA has an effect on HIV transcription. So 2 studies basically agreeing with each other, and so we hope to provide a third study here and verify those findings. I can tell you that there's a nice study that's also ongoing in Denmark by Thomas Rasmussen, and he's testing yet a third drug in this class. It's called panobinostat, and it's an HDAC inhibitor as well, and he's got some very nice data, and it seems like he's also seeing in some patients induction of HIV transcription resting cells. So these studies are going to be very informative, and so far it sounds like 2 of these drugs are are achieving what they're supposed to be achieving, or what we saw being achieved in vitro. So we hope that we may hit this first milestone, that there is hopefully a safe way to activate HIV transcription from resting memory T cells, which is the principal reservoir that we're all focused on.
And then there is a series of cascade of events or complementary or synergistic benefit to combining certain drugs, including maybe another class that has to do something else. You probably don't know where we're going to, you do know that there's probably going to be more than one compound involved. Is that true? Or?
Absolutely.
Yeah.
That's a great question,
Yeah.
actually. So what we do know already from some data that has been presented at this CROI meeting from John Meller's lab is these types of drugs, SAHA for example, only activates a small fraction of these latently infected cells that we've obtained from patients. So we know that we're going to need additional classes of drugs, and as you indicate, possibly acting synergistically. And how many, we don't know, but clearly there's going to be a need for additional classes of drugs to activate a greater proportion of that reservoir. So that seems like it's already quite obvious. Now, the challenge, of course, is to do this safely. If you start combining agents, you have to make sure that these are going to be agents that will not harm the patient. So that will be quite a challenge. We know how to do these things.
Tricky process.
Yep.
Stepwise.
Stepwise, very carefully done, making sure that first and foremost they're actually synergistic or additive in tissue culture or ex vivo, and then deciding whether these agents can safely be combined. So that's not trivial, and of course that's one of the goals.
Okay. So down the line, I think I mentioned to you that I had a conversation with Richard Klein and Kim Stribble from the FDA, and I said, God, we don't want to do an interview because we have no idea where we're going to go with this, and it's we conversed about that. This is a process. It's going to be like you said, tricky, and I think there will be probably they'll have to come up with a plan of trial design. They're talking about ethics, ethical informed consents, what this is going to mean, and I think maybe we should talk just a little bit about what we expect from patients. Is first of all, their patience. Because if we, the cure has been bandied about, and we will see news articles, unfortunately, which will say the cure is here and all kinds of things that are like it's in our presence at the moment. But it is a long process, and we anticipate and we hope that they will hold their excitement and their hope in a proper place and get into the trials altruistically or whatever is required, and make sure they understand the informed consents so that this isn't a surprise, that this is a long process. But we do need that support. We need the people who are living with AIDS to really come forward when the people like yourself and others in the ACTG feel it's necessary to conduct the trials. It's very important work, and we have to be on the same page together, I think. And but the programs that the FDA, I think, are going to be doing here this summer is some sort of an understanding of where the patients are, how to do, as you said, the process safely and well informed.
I agree, and just to illustrate the challenges that we're facing here, what I just described about SAHA and this interesting study of panobinostat in Denmark and soon our romidepsin study, the first goal is really to see whether we're activating expression of HIV in these reservoirs. The grand question is whether that reservoir is diminishing. It's one thing to activate expression of HIV, but if that reservoir is not declining, we will not be achieving our goal. So those clinical endpoints are much, much more difficult to assess. So we have a ways to go here before we can reliably determine whether that reservoir is diminishing. And of course, why this is important is because we need to figure this out before we stop antiviral therapy. As you know, if you stop antiviral therapy and there is no rebound, that would be a clear indication that we've achieved our goal. But we don't want to be doing this willy-nilly. Right. You need to have assays that are rigorous and that are predictive of the decline of this reservoir. That's the next big hurdle for us on the technical side. We need to achieve those assays. It's going to be very, very important. And so I know there's a lot of effort in the field going on in this area, because if we can do that, we can then predict maybe someone who has benefited from these treatments and has seen a reduction in reservoir and hence might merit an analytical treatment interruption, if you will, to see what happens to the viral rebound. So these are the next frontier. We're not quite there yet, but we're working very hard on these.
That was going to be my next question is what are your next steps? And that's beyond that, if there's anything else, we certainly like that. The one thing I do want to thank you for, not just you, but the companies that are engaged in this work, first of all, is working together and collaborating wherever it seems reasonable to do so and within the constraints of your proprietary issues, but also investing in the community's understanding and those advocates such as myself and many others who were able to review data confidentially so that there is a sense of transparency and that you really are moving forward in these directions. So I wouldn't be here with you if I didn't feel good about where we're at. I mean, that's my personal feeling. We did have that interview with Matt Sharp, Sharon Lewin, David Margolis, and Steve Deeks, which was a rich panel. And I think it's good to get that sense of where different people are with the stuff in your perception of your role and your interest in keeping people together and visiting and collaborating. It's a really good feeling, I have to tell you. And obviously you have doctors who are also advising and consulting with you all on sense of things as well. Is that true?
Yes, absolutely. and I must tell you, Fred, I've been doing work on HIV for 26 years now. So I've spent most of my adult life working on this problem and trying to solve it. And I must say, what I've seen lately with this eradication effort it's tremendous. I mean, the collaborative spirit is really outstanding. I can tell you that we have numerous collaborations with academics, with the government. As I mentioned, the ACTG was a great example, small companies. I mean, it's really tremendous how we're all coming together around this cause, and the collaborative spirit is fantastic. And I think, thanks to that, I'm hopeful that we'll accelerate our knowledge, and we'll hopefully accelerate our attainment of this cure, this functional cure that we're all so eager to be able to contribute to. So, but this collaborative spirit is really something special. And people that you mentioned, I know all of them very well. Terrific people. I work with them. In fact, I'm going to be giving a lecture with Matt in a few weeks at UC Berkeley. So yes, I'm very much involved with both the community, scientists and academia, other companies, governments. So it's really it's a wonderful atmosphere. It's the right atmosphere, and I'm optimistic that if there's something to be found, we will find this with this great collaborative effort.
I think it's worthy to mention the name of a person by the name of Martin Delaney,
yes,
who would be sitting here with us today, and he would be saying
he would be delighted to see what's happening. Right?
His sense would be that this is a very useful thing that you all are working together, and I think he would be certainly extremely pleased. And the thing that I'm mostly concerned about is that we really do have an energy from within, and also from organizations like the Forum, Veronica Miller's group, which is going to be doing some work with, I guess next June or so, they're planning another meeting. And these kind of collaborations are really exciting and really move things along. And I don't want to diminish the importance specifically of the cure, but the cure actually and the work that you're doing and the kind of work you're doing, the kind of collaborations, is a model for other diseases. I only hope that we, the companies that are also in other areas of research can say, hey, we ought to be doing this with cancer or with diabetes
It's a new paradigm, Fred. This is a new paradigm. This is not like, that's unlike anything we've ever done before. Right. You're absolutely right. And so I wonder if other areas will pick up on this.
I can't believe we'd be saying it here first, but if we are, I appreciate the opportunity to do that. But it seems like this is something that is so important, and we never were able to expand the kind of work that happens and the way it happens in AIDS to other fields of disease and chronic disease and acute disease, whatever it is. And I think that has to be, maybe top down, and we talked about a Manhattan Project for the cure, and I think in a sense we're in it.
It's happening. It's happening. That's exactly what's going on. Yeah. I mean, I can tell you, like I said, being very deeply involved in this area, I can see these collaborations, and really this exchange of information is fantastic. So it's really, I think we all realize that this is quite a daunting challenge, right. And we have to get together and basically work together to try to get to achieve this goal. So I think that's what's happening here.
Well, people like yourself, and I don't want to put myself in your shoes or anything, but I was there too at that last meeting, and that you were at to the very last presentation. Isn't that the way they do it? I mean, they suck you into this last meeting. And before I even say anything about that, we had Snowquester following sequester, that half of the docs left that were going northeast. They want to get out and back to their homes before the snow hit.
Right.
So even with that, the room was full.
Yep.
That particular room, a small room. But give us a little take on that. That was a very interesting
Yeah.
presentation.
So the last presentation was by our colleague Louis Picker, who works up at the Oregon Health Science University, and Louis has been doing some really phenomenal work, and he is basically using a CMV vector. So CMV is a herpes virus that a lot of people are infected with. It's benign. We don't know that we have it most of the time. But Louis has figured out a way to basically introduce, in this particular case, SIV, the HIV's cousin, SIV gene products into the CMV virus. So using a herpes virus to now put SIV genes and to use that to now instruct or immunize people later on. For now, it was monkeys. Immunize monkeys and demonstrate that he's able to achieve 50% protection upon challenge with an SIV strain. That's an aggressive SIV strain. So this is quite a remarkable achievement. And not only did he protect, but it seems like in his animals he achieved eradication of that virus. There's no virus to be found years after the vaccination and challenge. So this is a very interesting development and quite an exciting development. So the question obviously is, could this modality, this type of vaccination, work in a therapeutic setting? So if you basically have infected animals, or maybe down the road infected people, if you were then to vaccinate them after infection, could this modality help purge reservoirs, eliminate residual infection? Is unknown, and that's where the next step is going to be. But that data was quite exciting. I'm not going to go into the mechanistic details for this audience, but the breadth of CD8 responses that basically control SIV infection and also HIV was tremendous, and I think that has a lot to do with why that vaccine may have been as successful as it was. So that's the next frontier. This is again a very challenging area. CMV is not I mean it's a chronic virus. So to use this live attenuated chronic virus will have some issues, and we have to be very mindful of that and be very careful of that. But at this stage, it's a test of concept, and I think we're all eager to find out whether that type of a vaccination approach could help eradicate in a monkey an SIV infection that's already established. So very exciting. That's why that room was pretty full, because that data was pretty exciting and was pretty big news. And I congratulate Louis he has been working on this for 10 years, and it's quite an achievement. So let's see if we can now bring this to fruition and bring it to people, and whether it would work there is the question I think that the whole field has now.
I think obviously the room there would have been people in the hallway had it not been for Snowquester.
Of course, yes.
But I have to get back and put my political hat on for just a minute. I hadn't had the chance to do it, but we had the sequestration, which is I didn't realize how much it would impact, but 50% of the federal employees could not be here because they cut back on the sections, as it were. And they I'm sure they did so in a way that it impacted less than that they didn't just go by hierarchy. Some of the best or the top people who were there mostly to oversee and review and participate in other ways, meetings and so forth, were not able to come because they had people who were presenting, which was, of course, the optimal scenario. But to not have the people there to do the work, I know that it was a negative. And it's sad that we have to have this at a time where people, really need to interact with each other. This peer review process is very important to the development of everything, and it is the heart and soul of the research project. It's where we review everything. And we talked about a couple of the people and how they were young and how they were hit hard because they're young and we have to mold them into strong people that can stand up for their work. And that's part of it, I think, being able to get and present a whole bunch of data and then have people try to pick it apart and stand up.
Of course, like you said, the more people, the better. The more minds that are brought to this problem, the better. And it's a very important meeting for us annually. This is where we gather the best and the brightest people, and a lot of ideas are generated at these meetings. So indeed, it's a very important forum.
And one of the things that IFARA does always is the Young Investigators Panel. We had 4 very bright young investigators. Again, this is, I think, our 4th or 5th year we've been doing this here and at the IS. And I'm always inspired, as are the other doctors that go to the Young Investigators meeting that's prior to the CROI, to see that we are bringing in a very robust, bright group of young people who are going to take the place of, I mean, you're young yet, but there's a lot of docs who started in the field when AIDS GRID was first detected. And we're now down the road 30 years plus.
Yep.
And people are retiring. And I'm old enough to retire myself, but I guess I won't.
Yep. No, absolutely. I welcome the influx of new minds and
hopefully
fresh
you'll
minds.
be bringing them on board at Gilead as well.
Well, we'll see. Yeah.
Well, listen, Romas, I really appreciate you taking the time with me. It's always been a delight, and we look forward to more from you in the next meetings, and anytime you have a moment to spare with us, we'd be delighted to have
Always
you.
great to see you,
All right.
Fred. Always
Thank
great to
you,
talk
Romas.
to you.
Yeah.
Thank you for having me.
Yeah.